The title of my presentation is FDA Recommendations for COVID-19 Vaccine Development and Latest Updates. So this serves as an updated version of the presentation I gave at the New England Pharmacists Convention in September of this year. My name is Commander Paul Mouris. I am a Supervisory Investigator in FDA's Office of Regulatory Affairs, Office of Pharmaceutical Quality Operations 1. The objectives of my presentation are to 1. Describe how vaccines work to protect the body from disease. 2. Identify the realistic expectations for a safe and effective vaccine. 3. Recall the phases of vaccine development and the main purpose of each phase, including special considerations for a COVID-19 vaccine. 4. Outline the goals and structure of Operation Warp Speed. 5. Describe the potential role of accelerated approval and emergency use authorization for COVID-19 vaccine candidates. 6. List 5 of the leading COVID-19 vaccine candidates currently being developed and briefly describe the type of each vaccine candidate based solely on publicly available information. 7. Recall the storage controls required for the Pfizer and Moderna COVID-19 vaccines. So why are vaccines important? Vaccines protect against serious infectious diseases and save lives. The devastating effects of diseases such as polio, pertussis or whooping cough, rubella or German measles, and Haemophilus influenza type B are now rare due to vaccines. Infant deaths due to these diseases have essentially disappeared in the United States. Viruses and bacteria that cause vaccine-preventable diseases and death still exist and can be passed on to people who are not immunized. So how do vaccines work? Vaccines are designed to prevent infectious diseases or blunt the extent of illness. Vaccines work by mimicking infectious agents such as bacteria and viruses that cause disease and stimulate the body's immune system to build up defenses against them. Some vaccines contain weakened bacteria or virus, but it doesn't cause disease to the person. The disease-causing agents are foreign to the body and are called antigens. Vaccination exposes the body to antigens that are similar to the antigens found on the disease-causing agent. The vaccine prepares the immune system to respond quickly and forcefully if the body encounters the real antigen in the future. FDA serves as the gatekeeper for assuring the safety and effectiveness of vaccines. Safe refers to the relative freedom from harmful effect when prudently administered, taking into account the character of the product in relation to the condition of the recipient at the time. Pure refers to the relative freedom from extraneous matter in the finished product. And potent refers to the specific ability of the product to affect a given result. Vaccines must be manufactured consistently according to current Good Manufacturing Practice Regulations, or CGMPs. Some of the goals of vaccine development are to develop a rationale for the vaccine based on how the infectious agent causes disease, and identify what ingredients are needed for the vaccine, for example, the antigen, an adjuvant, or preservatives, and develop a manufacturing process for the investigational vaccine. Test the investigational product in animals for safety and effectiveness, i.e. preclinical studies, and then evaluate the investigational vaccine in human clinical trials. Ensuring safety of investigational vaccines. FDA evaluates sponsors' data to determine if it is reasonably safe to administer the investigational vaccine to people. Investigational vaccines cannot be administered to humans unless FDA gives the go-ahead. Vaccines intended for children are generally tested first in adults, with a step down to children later on. Vaccine candidates go through several phases of testing in people who volunteer to participate in clinical trials. FDA can stop clinical trials at any time if concerns arise. FDA sets the guidelines for the phases to assure the safety of volunteers, that good clinical practices are followed, and that the FDA's rigorous standards are met. Before an investigational vaccine sponsor can test the investigational vaccine in humans, they must first submit an investigational new drug application, or IND, to FDA. Once the IND is approved, the sponsor can begin Phase 1 clinical trials. Phase 1 clinical trials typically include 20 to 100 healthy volunteers for initial safety testing. There's an evaluation of the occurrence of adverse reactions, and the study may provide early evaluation of effectiveness, though the primary focus is on safety. Next comes Phase 2 clinical trials, and that typically involves several hundred volunteers. These are controlled clinical trials that provide additional information on safety, immunogenicity, and dose ranging. Sometimes sponsors will divide Phase 2 into 2A and 2B, where 2A focuses on safety and dose ranging, and Phase 2B looks at initial indications of efficacy. Then comes Phase 3 clinical trials, which typically involve thousands of volunteers. These are pivotal randomized controlled trials to determine efficacy, safety, and immunogenicity. The sponsor of an investigational vaccine would submit for approval from FDA by using a biologics license application, or a BLA. At the completion of various phases of clinical trials, companies may submit a marketing application called a BLA to FDA. The BLA contains all of the data gathered during preclinical animal studies and human clinical trials. The information in the application describes the vaccine's whole story, from product manufacturing and characterization, facilities where the vaccine was manufactured, data and results from the clinical trials, data and results from preclinical studies, the composition of the vaccine, and proposed labeling information for the healthcare provider and the patient. FDA reviews all of these data to determine whether the data support licensure of the vaccine for marketing authorization. FDA determines whether the vaccine is safe, pure, and potent, i.e., vaccine immunogenic or efficacious. Evaluation of non-clinical and clinical product used in development and proposed commercial scale manufacturing data and pharmacovigilance plans are submitted within the BLA. Vaccines and related biological product advisory committee review may also be used by FDA, especially if the vaccine represents a new technology or is a novel construct. The advisory committee makes recommendations to FDA not only on approvability, but also about indications and other specific questions posed to the advisory committee. The advisory committee consists of numerous experts in the field related to different areas of study. Typically, FDA will follow the advice of the advisory committee, but it is not obligated to. And then lastly, there are often pre-licensure inspections as part of the BLA review, where the FDA will inspect the proposed commercial scale manufacturer to assess whether or not it can handle the commercial scale manufacture thereafter. And then post-approval activities include lot release, annual or semi-annual inspections, adverse event surveillance, and Phase 4 studies. Phase 4 studies are studies that are required by FDA as a condition of approval to be conducted after approval. So the FDA scientific evaluation team, which reviews the BLA, includes clinical, CMC, assay, statistics, biomedical monitoring, pharm tox, pharmacovigilance, GMP inspection, labeling and promotion, communications, and others. So what does safe mean with respect to a vaccine? FDA takes vaccine safety very seriously throughout the vaccine life cycle. A vaccine is a medication or a drug, and like any other medication or drug, vaccines have benefits and risks. And although highly effective, no vaccine is 100% effective in preventing disease or 100% safe in all individuals. Millions of vaccine doses are given per year in the US, and the vast majority have few, if any, side effects. the benefits of vaccines clearly outweigh the potential risks. So what does effective mean? This is an important concept to understand with respect to human clinical trial endpoints for vaccines. Vaccine effectiveness is the assessment of whether a vaccine prevents disease in the general population. Vaccine development proceeds from a study of immunogenicity to a randomized controlled trial that determines vaccine efficacy under ideal conditions. Vaccine efficacy is defined as the reduction in the incidence of a disease among individuals who have received a vaccine compared to the incidence in the unvaccinated people. Efficacy of a new vaccine is measured in phase 2 or phase 3 clinical trials by giving one group a vaccine and comparing the incidence of disease in that group to another group who did not receive the vaccine, or typically received a placebo. Vaccine effectiveness depends upon vaccine efficacy. So the benefits and risks. Science provides FDA with the information, but science does not provide all of the answers. FDA expertise is utilized to make decisions based on the benefits and risks for a population. FDA evaluates the benefits and risks for the population. The healthcare provider evaluates the benefits and risks for a patient, and the patient evaluates the benefits and risks in terms of their personal values. Vaccine ingredients. A vaccine is made up of various ingredients, and each ingredient present in a vaccine is there for a specific reason. Different ingredients have different roles in a vaccine, and vaccines licensed for use in the United States are demonstrated to be safe and effective before they are used by the public. So it's not just the antigen present in the vaccine that's intended to stimulate the immune response. There's also preservatives to maintain sterility potentially, and then sometimes there are adjuvant agents which are used to aid the immune system response. With respect to vaccine manufacturing, once a vaccine is licensed, the manufacturing facilities used are inspected by FDA approximately every 2 years, or more or less frequently based on FDA's internal risk assessment of that facility. Experienced FDA investigators carefully examine and evaluate the operation for compliance with FDA regulations and good manufacturing practice regulations. This is actually my role as a lifetime FDA investigator and current supervisory investigator within Office of Regulatory Affairs. Manufacturing processes for vaccines are very complex. Each product lot or batch is tested by the manufacturer for purity, potency, identity, and sterility. FDA requires vaccine manufacturers to submit protocols of all processes, procedures, and standards with results of all applicable tests, along with samples from the vaccine lot to FDA to assess for lot release. Manufacturers are not permitted to distribute a lot of a licensed vaccine until FDA releases that lot. There's also post-approval monitoring of vaccines, which largely assesses the ongoing safety and adverse reactions and continued efficaciousness of the vaccine. FDA's work continues long after vaccine approval. There's intense oversight of the production of vaccines after licensure to ensure continuing safety. Vaccine Adverse Event Reporting System, or VAERS, is co-administered by FDA and CDC. Collaboration with CDC on a program called Vaccine Safety Data Link also is used to monitor vaccine safety after approval. Collaboration with other U.S. government agencies also occurs, such as Centers for Medicare and Medicaid Services, DoD, the Veterans Administration, and Indian Health Services, to name a few. Continued international collaboration occurs for vaccine monitoring as well. Information sharing agreements with our counterparts in other countries, as well as the World Health Organization. And in 2007, the FDA Amendments Act included something called the FDA Sentinel Initiative, which is a large surveillance national electronic data system for monitoring adverse events and signals of potential adverse events or issues with approved drug products. It strengthens FDA's ability to monitor the safety of medical products writ large. And there's also the PRISM program, the Post-Licensure Rapid Immunization Safety Monitoring, for the monitoring of vaccines and other drug products after approval. It was established during the H1N1 outbreak and used active safety surveillance. It evolved into mini Sentinel pilot projects, expanding capacities to general vaccine safety monitoring and evaluation. So this brings us to quiz question number 1, and it's a true-false question. Vaccine manufacturers can distribute manufactured lots of vaccines after each lot passes all internal release testing requirements. Is this true or false? I'll give you about 20 seconds. The answer is false. Unlike for traditional drugs where manufacturers themselves make the final decision to release lots manufactured, vaccine manufacturers must send all their internal release testing results and samples from manufactured vaccine lots to FDA, and only FDA can formally release the vaccine lot for distribution. So in June of 2020, FDA released a new guidance for industry entitled Development and Licensure of Vaccines to Prevent COVID-19. This guidance describes FDA's current recommendations regarding the data needed to facilitate clinical development and licensure of vaccines to prevent COVID-19. A significant portion of the guidance is dedicated to assessing the vaccine-associated enhanced respiratory disease potential of the investigational vaccine. Data from studies in animal models administered certain vaccine constructs against other coronaviruses, namely SARS and Mers, have raised concerns of a theoretical risk of COVID-19 vaccine-associated enhanced respiratory disease. In these studies, animal models were administered vaccine constructs against other coronaviruses and subsequently challenged with the respective wild-type coronavirus. Evidence was observed of immunopathologic lung reactions characteristic of a T helper cell type 2 hypersensitivity, similar to enhanced respiratory disease described in infants and animals that were administered a formalin inactivated RSV vaccine and then subsequently challenged with RSV. So key considerations for clinical trials. Understanding the SARS-CoV-2 immunology and specifically vaccine immune responses that might predict protection against COVID-19 is currently limited and evolving. Thus, while evaluation of immunogenicity is an important component of COVID-19 vaccine development, at this time the goal of development programs should be to pursue traditional approval via direct evidence of vaccine efficacy in protecting humans from SARS-CoV-2 infection and/or disease. Clinical development programs for COVID-19 vaccines might be expedited by adaptive and/or seamless clinical trial designs that allow for selection between vaccine candidates and dosing regimens and for more rapid progression through the usual phases of clinical development. So clinical trial design. Early phase trials often aim to down-select among multiple vaccine candidates and/or dosing regimens via randomization of participants to different treatment groups. While including a placebo control and blinding are not required for early phase studies, doing so may assist in interpretation of preliminary safety data. Later phase trials, including efficacy trials, should be randomized, double-blinded, and placebo-controlled. To ensure that a widely deployed COVID-19 vaccine is effective, the primary efficacy endpoint estimate for a placebo-controlled efficacy trial should be at least 50% relative to the control group. Phase 3 clinical trial endpoints. Either laboratory-confirmed COVID-19 antibody test or laboratory-confirmed SARS-CoV-2 infection is an acceptable primary endpoint for COVID-19 vaccine efficacy trials. FDA recommends that either the primary endpoint or secondary endpoint, with or without formal hypothesis testing, be defined as virologically confirmed SARS-CoV-2 infection with one or more common flu-like symptoms or new loss of taste or smell. As it is possible that a COVID-19 vaccine might be much more effective in preventing severe versus mild COVID-19, sponsors should consider powering efficacy trials for formal hypothesis testing on severe COVID-19 endpoints. So this brings us to quiz question number 2. To ensure the COVID-19 vaccine candidate is effective, the primary efficacy endpoint estimate for a placebo-controlled efficacy trial should be at least A, 100%, B, 90%, C, 60%, or D, 50%. And again, we'll give you about 20 seconds. And the answer is D, 50%. So this may seem like a low bar, but recall from slide 11 that vaccine effectiveness and efficacy only includes the percent in which the disease is altogether prevented. So it does not include the percent who had the disease in a less severe form as a result of being administered the vaccine. So what role might accelerated approval play with COVID-19 vaccine development and approval? Accelerated approval of a new drug application or a new biologics license application is when FDA authorizes the use of acceptable surrogate endpoints to measure drug vaccine efficacy in lieu of direct evidence of the intended clinical endpoint. So an example might be lowered LDL as an endpoint in lieu of reduced heart attacks or cardiovascular related deaths for statin drugs. However, there are currently no accepted surrogate endpoints that are reasonably likely to predict clinical benefit of a COVID-19 vaccine. For that reason, at this time, the goal of development programs should be to pursue traditional approval via direct evidence of vaccine safety and efficacy in protecting humans from SARS-CoV-2 infection and/or clinical disease. Another potentially important pathway for making a COVID-19 vaccine available to the public is the emergency use authorization. You may have heard of FDA granting emergency use authorization to COVID-19 treatments such as remdesivir and convalescent plasma. An emergency use authorization may be issued only after several statutory requirements are met. Among these requirements is a determination by FDA that the known and potential benefits of a product when used to diagnose, prevent, or treat serious or life-threatening diseases outweigh the known and potential risks of the product. Thus, for a vaccine for which there is adequate manufacturing information, issuance of an EUA may be appropriate once studies have demonstrated the safety and effectiveness of the vaccine, but before the manufacturer has submitted and/or FDA has completed its formal review of the biologic license application. So that brings us to the federal program entitled Operation Warp Speed. It's an unprecedented public-private partnership with the common goal of making a safe and effective COVID-19 vaccine available by January of 2021 and accelerate other treatments and diagnostics for COVID-19. It's a collaboration of the private sector and several U.S. federal government departments, including Department of Health and Human Services, Department of Defense, U.S. Department of Agriculture, Department of Energy, and the Veterans Administration, among several others. The strategy is for one, to release the strategy for distributing a COVID-19 vaccine, to release the CDC COVID-19 vaccination program interim playbook, and then the vaccine will be reviewed under FDA standards per the June 2020 guidance. And then CDC's Advisory Committee on Immunization Practices will independently review and make vaccine administration recommendations. So this very informative slide on Operation Warp Speed's accelerated vaccine process really shows and highlights the difference between the traditional development and review process, which generally takes on average about 73 months to complete, compared to the COVID-19 accelerated process under Operation Warp Speed, which it proposes will be about 14 months. And that is, in fact, about what it's been to get to the point where we have an authorized vaccine through emergency use authorization. Of course, we definitely do not have the 300 million doses so that every American can get vaccinated, and probably won't for to the middle of next year at earliest. But some things to note on this slide are the R&D and phase 1 clinical trials, which are typically done sequentially, sort of being done simultaneously for that 5-month time period during the accelerated process, as are phase 2 and phase 3 clinical trials in the accelerated COVID-19 vaccine development done simultaneously. And then you'll also notice that the brown section of the chart represents manufacturing. Typically that wouldn't take place until after approval. But for the leading COVID-19 vaccine candidates, they have been manufacturing all along to ensure that immediately after emergency use authorization is granted, they have millions of doses available to immediately distribute. So now we're going to discuss the types of COVID-19 vaccines currently going through the development process. So the first category of COVID-19 vaccines we're going to discuss are the messenger RNA-based vaccines. The messenger RNA contained in these vaccines instructs our body's cells to produce the SARS-CoV-2 spike proteins to elicit an immune response. And these are the proteins that protrude from the surface of the virus that would typically be recognized as an antigen by our immune system and generate an immune response. So examples of this type of vaccine include Moderna's COVID-19 vaccine, as well as the Pfizer-BioNTech COVID-19 vaccine in development. And perhaps one you might not have heard of is a German company called CureVac. They also have a messenger RNA-based COVID-19 vaccine in development. And this vaccine reportedly has the financial backing of Tesla's Elon Musk. So messenger RNA vaccines have benefits and risks which are pretty significant. The advantages are that messenger RNA vaccines are cheaper and can be manufactured much faster than a protein-based vaccine. So they're highly scalable to make millions of doses very quickly. They don't have to manufacture the protein, which is always rate limiting, as the messenger RNA instructs the cells of our body to make the viral proteins. So our bodies become sort of the manufacturer of the spike protein. But the risks are quite significant in that no messenger RNA vaccines have ever been used before. So the risk of failure is higher with mRNA vaccines when large numbers of subjects are exposed to this type of vaccine. They may also be more likely to produce flu-like symptoms than traditional vaccines. So the next category of COVID-19 vaccines we're going to discuss are the adenovirus vector-based vaccines. The one you've probably heard about the most is the AstraZeneca University of Oxford COVID-19 vaccine, which uses a chimpanzee adenovirus called Oxford-1, which has been genetically modified to produce SARS-CoV-2 antigens. And then a Chinese company called CanSino Biologics has a COVID-19 vaccine, which uses adenovirus, which has been genetically modified to express SARS-CoV-2 proteins as well. And Janssen has a COVID-19 vaccine development, which actually uses a genetically modified human adenovirus called AdVac. So here are some examples of other types of COVID-19 vaccines in the development pipeline. Another Chinese company called Sinovac Biotech has a coronavac vaccine which uses SARS-CoV-2 virus obtained from actual infected hospital patients, which they grew in cell lines and then chemically inactivated. Sanofi Pasteur is teaming with GSK and repurposing its earlier SARS vaccine efforts into a COVID-19 vaccine. Its vaccine candidate uses recombinant DNA technology, which is a platform they already use for their approved flu vaccine called Flublok. And then Novavax has a COVID-19 vaccine which uses SARS-CoV-2 spike proteins packaged into nanoparticles to elicit an immune response. So now we're going to go into a little more detail regarding the leading vaccines, which are probably closest to obtaining emergency use authorization, and some of the logistical obstacles that you as pharmacists will probably have to improvise or overcome with respect to these vaccines. So the two leading candidates are Pfizer and Moderna, as you know. Both are messenger RNA based vaccines, which, as we discussed, provide instructions to our cells to make viral protein that can be recognized by the immune system and elicit an immune response. The Pfizer vaccine, as you've probably heard in the news, has to be stored at ultra cold temperatures, minus 80 degrees C, minus 94 degrees Fahrenheit. The Moderna vaccine actually can just be stored in a simple freezer down to minus 20 degrees C, but it's also stable in traditional refrigerated temperatures 2 to 8 C for up to 30 days. So it has a distinct advantage logistically over the Pfizer vaccine in that regard. So 5 of the 6 early vaccine candidates are 2-dose series, so that's something to consider as well. Patients who get a single dose would have to come back and be tracked in some way to get their second dose 3 to 4 weeks later. 1 of 6 early vaccines is stored at refrigerated temperatures, only 1 of 6. 1 is stored at ultra cold temperatures, obviously the Pfizer, and then 4 can be stored in a regular freezer. So the storage of these vaccines is definitely something that logistically will have to be planned and thought out in advance at your pharmacies and hospitals. So again, the majority of the leading vaccine candidates are 2-dose series, which must be given 21 to 28 days apart. So providers must plan for timely second dose of the same brand of the vaccine within that time period, and perhaps set up systems for reminders to patients to come back in and tracking not only when they got their first dose, but which brand they got once we start getting multiple vaccines available to us. The brands are not interchangeable. In early phases of the vaccine distribution, Operation Warp Speed will reserve supply of the second dose of the vaccine. And co-administration of other vaccines was not studied, therefore it's unlikely to be recommended. So you'll have to separate, for example, the flu vaccine from the COVID vaccine, by a matter of 2 or 3 weeks, or whatever CDC recommends. And then the duration of protection is unknown at this time. So that will continue to be studied. But for now, we don't know for sure how long protection will last. So here are some of the details about the Pfizer-BioNTech vaccine that have been published to this point. From phase 3 clinical trials and also phase 1 and 2. The tolerability is determined to be well tolerated. Of course, that's subjective, but mostly they've seen mild to moderate side effects like soreness at the injection site, fever, chills, headache, and muscle or joint pain. So sort of common side effects that you see from vaccine administration. And the neutralizing titers that they've seen have been 1.9 to 4.6 times higher than serum plasma for patients diagnosed with COVID-19 disease. And that would be after the second injection. Obviously, the titers will be much lower after only the first injection, though still there is some protection provided. And then the efficacy, which was announced on November 18th, initially it was announced that it was over 90% effective. And then about a week later, they came out and said that it was actually 95% effective in the second analysis. And that was an independent data monitoring committee. And the sponsor has applied for EUA. So here are some of the logistics on the Pfizer-BioNTech vaccine, and really what dominates the slide are the storage requirements, which are really logistically prohibitive for many sites that just don't have the capacity to store vaccines at minus 60 to minus 80 degrees C. So the company is going to try to make it as painless as they can. They're going to be shipped to the site directly from the manufacturer in ultra cold shippers. The recharge shipper with pelletized dry ice within 24 hours of receipt. The first recharge of dry ice will be sent to the site at no cost by the government. And they should be replenished with dry ice every 5 days thereafter to maintain the required temperature. The total amount of pelletized dry ice needed per thermal shipper is approximately 23 kilograms. And on day 15, should transfer the vaccine to refrigerated temperatures of 2 to 8 degrees C, and then use within 5 days of transferring to the refrigerator. Shippers may only be opened 2 times a day and ideally less than 3 minutes each time. And shippers will come with data loggers and will need to be returned to the manufacturer within 20 days of receipt. So here are some important logistical timeframes to understand with respect to the stability of the Pfizer vaccine. It can be stored in the transport shippers provided by Pfizer for up to 15 days with dry ice recharges, after which time it should be transferred to a refrigerator. And then it can be stored in the refrigerator for up to an additional 5 days. So if you consider that the total storage time from a transport shipper is 20 days, so it is not likely that you're going to be able to get a first and second dose out of that first shipper. You're probably going to have to wait on a second shipment to get the second dose for a patient. So each shipment would essentially just be a single dose per patient. It could be stored in an ultra cold freezer if a facility actually has one for up to 6 months. And then it can be stored at room temperature for 2 hours and then 6 hours after dilution. And it'll be available in increments of 975 doses. And each shipper will contain 5 trays of vaccines. And there'll be 5 doses per vial, and they'll require dilution. And again, the dosing schedule will be 2 dose series at 0 and 21 days. So the second leading COVID-19 vaccine candidate is the Moderna messenger RNA vaccine. And in terms of tolerability, similar side effects were seen with this vaccine: pain at the injection site, fatigue, muscle pain, headache, and chills. And side effects were found to be more common after the second dose in the 2-dose regimen. And neutralizing titers were higher than serum plasma for patients diagnosed with COVID-19 disease. I didn't see consistent numbers, in my research, but I think one thing it's important to note is that it's sort of apples to oranges when looking at Pfizer's neutralizing titers compared to Moderna's, because the nominal titer that they were comparing their vaccine titers to was not the same. So they didn't have the same makeup of patients who recovered without ever having to be hospitalized, didn't have what would be called severe disease. There are different ratios of the number of patients they used to represent sort of the nominal titer. So that's something to keep in mind. And then on November 16th, 2020, they announced that their efficacy was determined to be 94.5% based on the initial analysis. So here are the logistics for the Moderna COVID-19 vaccine, and what immediately jumps out at you is that it can be stored at minus 20 C or closer to traditional freezer temperatures. And one might ask, why would two messenger RNA vaccines have to be stored at such drastically different temperatures? And while we don't know the answers to that, a lot of it's proprietary. From what I understand, it has to do with the liposome that the messenger RNA is contained within, sort of like lipid nanoparticles. For the Moderna vaccine, apparently that liposome is more protective and more hardy. This is somewhat speculative as we don't know the details because, again, it's proprietary. But I'm sure Pfizer would love to know how Moderna was able to have their vaccine stable at only minus 20 degrees C. So it'll be shipped to the site from McKesson, similar to the varicella vaccine, and it'll be stable for 30 days once taken to refrigerated temperatures of 2 to 8 C, and then stable at room temperature for up to 12 hours. It'll be available in increments of 100 doses. That's 10 multi-dose vials, which is 10 doses per vial, obviously. And it actually does not require dilution or mixing. And like the Pfizer vaccine, it'll be a 2-dose series, except that it'll be at 0 and 28 days rather than 0 and 21 days. AstraZeneca recently announced that their COVID-19 vaccine produced 70% efficacy. But the backstory of how they arrived at that number is actually quite fascinating. This adenovirus-based COVID-19 vaccine was intended to be given in a 2-dose regimen 28 days apart. But due to an alleged manufacturing error, some study participants, approximately 2,700, received a half-strength dose for their first of 2 doses, followed by a full-strength dose for their second dose. And then the intended 2 full-strength dose regimen demonstrated 62% efficacy in approximately 11,000 participants in phase 3 clinical trials. The accidental half-strength dose followed by the full-strength dose regimen actually resulted in 90% efficacy. So the firm reported a combined efficacy of 70%, combining those 2 pools. It is not clear yet if the 90% efficacy regimen for the group accidentally receiving the low first dose is a statistical anomaly or if it's statistically significant. But it should be noted that AstraZeneca's COVID-19 vaccine can be stored at refrigerator temperatures, so this could prove a significant logistical advantage over the messenger RNA vaccines that are likely to be authorized ahead of it. And to put that in context, even if the AstraZeneca efficacy was 62%, which it demonstrated to be in the 2 full dose regimen, that would still be the approximate efficacy of the nominal flu vaccine that we're given each year. If you recall from earlier in the presentation, the FDA reported standard is 50% efficacy. So 62% is certainly enough to get emergency use authorization and or approval down the road, but just compared to the 94.5 and 95% efficacy reported by the 2 messenger RNA-based COVID-19 vaccines, it just seems to pale in comparison. But just to put that into context. As you likely heard, the Vaccine and Related Biological Products Advisory Committee met on December 10th to deliberate on the Pfizer-BioNTech COVID-19 vaccine emergency use authorization request. The committee voted 17-4 that FDA should provide emergency use authorization to the Pfizer-BioNTech COVID-19 vaccine for those 16 years old and older, thus paving the way for FDA to grant emergency use authorization to the Pfizer-BioNTech vaccine. This same committee will also hold another meeting on December 17th of 2020 to review and vote on Moderna's COVID-19 vaccine emergency use authorization request. This slide represents an up-to-date list of authorized COVID-19 vaccines around the world. China gave emergency use authorization to their Sinovac and CanSino COVID-19 vaccines back in July of 2020. They have already vaccinated thousands of their citizens and have started exporting these vaccines to other countries, such as Indonesia. The UK granted emergency use authorization to the Pfizer-BioNTech COVID-19 vaccine back on December 2nd and have started vaccinating their healthcare workers and nursing home residents as of December 7th. Canada authorized the Pfizer-BioNTech COVID-19 vaccine on December 9th under interim order. And on December 11th, Saudi Arabia and Bahrain granted emergency use authorization to the Pfizer-BioNTech COVID-19 vaccine as well. So I should say that there is one quote-unquote approved COVID-19 vaccine, and that is the Russian vaccine called Sputnik 5, formerly known as GAM-COVID-Vac. It was developed by Gamaleya Research Institute in Moscow. It was approved by the Ministry of Health of the Russian Federation on August 11th of 2020. However, experts have raised considerable concern about the vaccine's safety and efficacy, given that it has not yet entered phase 3 clinical trials at the time that approval was granted. One of the most important questions that need to be asked and resolved during this pandemic as vaccines become available is how to deploy approved and authorized COVID-19 vaccines. The federal government has already signed contracts in the billions of dollars with various investigational COVID-19 vaccine sponsors for tens of millions of doses, including Pfizer, Moderna, and AstraZeneca. But how will vaccine doses be distributed and administered at this scale? Who will get vaccinated first as vaccines become available? Recent development on December 7th, Connecticut Governor Ned Lamont issued Executive Order 9Q, which authorizes qualified Connecticut pharmacists to administer authorized COVID-19 vaccines and approved flu vaccines to all patients over the age of 10. So clearly, pharmacists are going to play a very significant role in the administration of vaccines at this incredible scale that's going to be required in the coming months. Many federally subsidized COVID-19 testing centers will be converted to drive-through vaccine administration centers, but the need to monitor patients for adverse reactions and fainting will require post-vaccination monitoring at these drive-through vaccine administration centers. On December 3rd, 2020, the CDC's Advisory Committee on Immunization Practices published their interim guidance on COVID-19 vaccine distribution. The committee recommended as interim guidance that both healthcare personnel and residents of long-term care facilities be offered the COVID-19 vaccine in the initial phase of the vaccination program rollout. There are approximately 21 million U.S. healthcare personnel working in settings such as hospitals, long-term care facilities, outpatient clinics, home health care, public health clinical services, emergency medical services, and pharmacies. There are approximately 3 million adult residents at long-term care facilities, which include skilled nursing facilities, nursing homes, and assisted living facilities. So this slide is intended to demonstrate what COVID-19 vaccine distribution might look like adjusted as volume of the vaccine increases over time. So in the initial phase, where we have the first vaccine becoming available, it would be mostly healthcare practitioners and first responders, plus people with high-risk conditions, older adults, including those living in long-term care facilities. And then as larger number of doses become available, we would expand to non-healthcare critical workers, people in congregate settings, and all other older adults. And then furthermore, we could get into young children and other critical workers. Now, the vaccine has not been studied, or many of these vaccines have not been studied in children younger than the age of 16. So that could complicate things, and it may be that parents might not feel comfortable giving the vaccine to children younger than that until more is known. There are studies ongoing in younger children, but none of them have published any data that you could sort of hang your hat on. And then there'd be continued vaccination. There'd be a shift in strategy. So once we sort of reach herd immunity, all others in the U.S. who did not have access in the previous phases would get vaccinated. And one thing that we've seen in the U.K., the Pfizer vaccine being given, we've seen folks that are prone to allergies have some significant allergic reactions. So what they're saying out of the UK is that folks that are known to be allergic and have significant allergic reactions to other medications and other things should probably hold off and not get the vaccine, at least initially, until more is known and more is understood. So this concludes my presentation, and I wanted to thank you all for your time and attention.